Hi, my name is Aaron, and I will be going over the Kreb cycle, what it is, and how it works. The human body is an amazing instrument that allows us to do many complex activities such as playing basketball, climbing up a mountain, and creating YouTube videos for a cell biology class. All of these activities, however, do require energy. As humans, we cannot obtain our energy directly from the sun like plants do, but indirectly by eating food such as carbs, proteins, and fats. Our bodies convert these foods into energy in the form of ATP through a complicated series of metabolic pathways. The first step in obtaining this chemical energy is done by the digestive system, which breaks down the food into usable molecules. From here, these molecules are ran through a series of interactions that produce the ATP our cells need to survive. For example, let's consider the breakdown of glucose. Glucose, a 6-carbon molecule, is broken down into 2 pyruvate molecules, 3 carbons apiece, in a process known as glycolysis. Glycolysis occurs in the cytoplasm of eukaryotic cells and produces a net total of 2 ATP and 2 NADH molecules. After this, pyruvate must be oxidized by pyruvate dehydrogenase, which produces NADH and acetyl-CoA. Which brings us to the Kreb cycle. The Kreb cycle, named after the man who discovered it, Hans Krebs, is a central metabolic pathway that generates energy through the oxidation of acetyl-CoA. The Krebs cycle, or citric acid cycle as it is sometimes called, takes place within the outer matrix of mitochondria and eukaryotic cells and produces a total of 3 NADH, 1 FADH2, and 1 ATP per acetyl-CoA molecule. The cycle consists of 8 enzyme-catalyzed reactions, beginning with a reaction between acetyl-CoA and oxaloacetate, producing citrate. This reaction is catalyzed by the enzyme citrate synthase. In step 2 of the cycle, citrate is converted to isocitrate, which is catalyzed by the enzyme aconitase. The enzyme isocitrate dehydrogenase catalyzes the third reaction, which converts isocitrate to alpha-ketoglutarate and also produces a molecule of NADH. In step 4, alpha-ketoglutarate reacts with its enzyme alpha-ketoglutarate dehydrogenase to produce succinyl-CoA. A molecule of NAD+ is reduced to NADH in this step also. In step 5 of the cycle, succinyl-CoA interacts with the enzyme succinyl-CoA synthetase in a reaction that produces succinate. A molecule of GDP is phosphorylated to produce GTP in this step as well. Step 6 sees the enzyme succinate dehydrogenase catalyze the reaction that converts succinate to fumarate. An FADH2 molecule is produced in this step. Next, in step 7, fumarate and a molecule of water react to form malate in a reaction that is catalyzed by fumarase. Finally, in step 8, oxaloacetate is produced by the reaction between malate and the enzyme malate dehydrogenase. This step also produces an NADH. With oxaloacetate regenerated, the Krebs cycle is complete and ready to begin again when another acetyl-CoA molecule is provided. Combined, glycolysis and the Krebs cycle produce a net total of 4 ATPs, 10 NADHs, and 2 FADH2s per molecule of glucose. The NADH and FADH2 molecules are passed on to the electron transport chain, where they produce an additional 34 ATPs. This means, in an ideal situation, our bodies will produce 38 ATPs per molecule of glucose. I hope this video helped you understand a little more about the Krebs cycle than you already did. Thanks for watching.